in HIV-infected patients. As introduction, we know that HIV infection remains a public health problem. From WHO report, there were 37.6 million persons living with HIV worldwide in 2020. Infected persons, treated or not by ACT, have often metabolic disorders such as dyslipidemia, a risk factor for cardiovascular diseases. However, all these patients do not develop dyslipidemia, raising the suspicion that host-related genetic predisposing factors influence the occurrence of dyslipidemia. Several single nucleotide polymorphisms are associated with the occurrence of this disorder, particularly in apoprotein genes. It has been shown that HIV-associated dyslipidemia is accompanied by an increase in lipoproteins containing apoprotein B, particularly LDL cholesterol, which increase is known to be one of the predictive factors of the cardiovascular risk. Thus, the apoprotein B100 gene is one of the most studied genes of interest, particularly the EcoR1 polymorphism at position 4154, a missense mutation. This guanine to adenine substitution implies the loss of the restrictive site of this endonuclease and the change of amino acid from glutamic acid to lysine. The aim of this study was to establish the link between EcoR1 polymorphism in exon 29 of the ApoB100 gene and changes in serum lipoprotein profile in ART-naive patients living with HIV in Ivory Coast. About the material and method used, the study population consisted of HIV-negative controls who were voluntary blood donors and persons living with HIV monitored at the National Blood Transfusion Center. All of them presented at least one disturbed lipid report in classical lipidemia and apoproteinemia. The laboratory for the biological analysis was the CEDRES laboratory. The primers pair used was named BC2 and BC2R with sequence of interest on exon 29 of ApoB100 gene. For the digestion step, we use the restrictive enzyme endonuclease EcoR1. We performed a cross-sectional study. Whole blood collected on EDTA and dry tubes was sent to CEDRES where aliquots were done, stored at -80 degrees Celsius, and then lipoprotein parameters were analyzed. Sociodemographic, therapeutic, and biological data were collected from the database of the first part of the study. Indeed, the first part of the study had already been carried out using these same samples in 2013. In order to determine the EcoR1 polymorphism, we performed PCR digestion and DNA electrophoresis. All recruited persons gave the informed consent. The protocol was approved by the National Ethics and Research Committee. The collected data were analyzed using SPSS software. The presence of DNA digestion was noted plus and its absence minus. The comparison were made using student tests for quantitative values and Q-square or Fisher-exact tests for qualitative values. The tests were considered significant at the risk 5%. The Adi-Van Baer equilibrium was tested for allele distribution interpretation, and the significant level was 3.84. A total of 55 subjects were recruited, consisted of 42% of HIV-naive persons living with HIV, and 58% of HIV-negative control. According to the sociodemographic cardiovascular risk factors, there was a male predominance in both groups, but there were no significant differences regarding gender and age distribution. The mean body mass index was higher in persons living with HIV than in HIV negative. The values were 24% versus 26, and the p-value was 0.046. Also, high body mass index values were more frequent in HIV-infected people than HIV negative. The values were 63.6 versus 35.5, with p-value 0.043. According to serum lipid and apoprotein concentration levels, apoproteinemia A1 was more frequent in persons living with HIV, and the p-value was 0.002. There was in both groups the predominance of hypertriglyceridemia, hyperapoproteinemia B, hyperapoproteinemia Lp(a), as well as high heterogeneity indexes for both ratios. But the difference was significant only for the heterogeneity index, defined by the ratio total cholesterol divided by HDL cholesterol. The p-value was 0.023. According to the EcoR1 polymorphism, the wheel genotype predominated in both groups, and all three genotypes were found in both. The wheeled allele predominated in HIV-negative subjects, while the mutant one predominated in persons living with HIV. P-value was 0.002. The Ardevine-Berg equilibrium were deviated in both HIV-negative and persons living with HIV subjects. The p-value were respectively 0.008 and less than 0.0001. About the distribution of genotypes and alleles according to the demographic cardiovascular risk factors, regardless of HIV status, genotype frequencies were not at Ardevine-Berg equilibrium for any of the demographic factors. The mutant allele was more frequent in those under 40 years of age, women, and those with high body mass index values. According to HIV status, in the HIV-negative group, Ardevine-Berg equilibrium was deviated in those under 40 years of age, male, and those with high body mass index. In persons living with HIV, the equilibrium was deviated in all subgroups with frequencies of a mutant allele in proportion higher than 10%. Studying the EcoR1 polymorphism according to serum lipid and apoprotein levels, regardless of HIV status, only the genotypic frequencies in subjects with high LDL cholesterolemia were at Ardevine-Berg equilibrium. The mutant allele was more frequent in subjects with increased triglyceridemia, LDL cholesterolemia, apoproteinemia Lp(a), heterogeneity index total cholesterolemia divided by HDL cholesterolemia, and apoproteinemia B100. In those with normal total cholesterolemia and in those with decreased HDL cholesterolemia and apoproteinemia A1. Taking HIV status into account, the mutant allele was more frequent in persons living with HIV than in HIV-negative subjects. The values were respectively 40 to 32% and 2 to 23%. Concerning the lipid concentration, the mutant allele was more frequent, especially in persons living with HIV, presenting total hypercholesterolemia 28.1%, normal LDL cholesterolemia 26.7%, HDL hypocholesterolemia 27.3%, hyperapoproteinemia LP small a 28.1%, and high heterogeneity index as total cholesterolemia divided by HDL cholesterolemia 23.7%. For apoprotein concentration, the mutant allele was more frequent, especially in persons living with HIV, presenting hypoapoproteinemia A1 32.1%, hyperapoproteinemia B100 28.1%, and high heterogeneity index as apoproteinemia B divided by apoproteinemia A1 33.7%. In conclusion, the results observed in the general population differed from those in the person living with HIV group. The distribution of alleles was not random, and the mutant allele was more frequent in persons living with HIV. And this exposed them to increased body mass index, atherogenic apolipoproteins, and heterogeneity index, and exposed them also to decreased protective apolipoproteins. As perspective, in order to investigate the link between the effect of ART treatment, apolipoprotein B100 and A1 genes variation, we are considering doing analysis on a population of persons living with HIV on ART treatment. Thank you for your attention.